Hi, everyone. This is your People Behind the Science podcast host, Dr. Marie McNeely, and I am thrilled to have you here with us to celebrate our 850th episode with our guest, Dr. Dave Jackson. Listeners, if you want to learn more about our show, check out past episodes, find our recommended book lists, and more, visit us at peoplebehindthescience.com. And in this episode, Dave shared some excellent stories, so I hope you're ready to meet another one of our brilliant people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am thrilled to be speaking with our guest researcher, Dr. Dave Jackson. So, Dave, welcome to the show today. How are you?
I'm doing great, thanks. Excited to be here.
Well, Dave, we're excited to learn more about you, your work, and your career. But before we get into the details, let me first take a moment to tell our listeners a little bit more about your current position and how you got there. So listeners, Dave is professor of plant genetics at Cold Spring Harbor Laboratory. He obtained his bachelor's degree in biotechnology from the University of Leeds, and he was awarded his PhD from the John Innes Institute. Afterwards, he conducted research as a NATO and USDA postdoctoral fellow at the Plant Gene Expression Center in Berkeley, California, before joining the faculty at Cold Spring Harbor in 1997. In 2018, Dave was elected as a fellow of the American Association for the Advancement of Science. And Dave, today we're excited to know more about some of your accomplishments, your research, and your career. But we also want to know more about your life outside the lab. So can you tell us, what do you like to do when you're not doing science?
Too many things. I think one of the great challenges in life is to have a balance between work and things outside work. And that's certainly true in science. But I really enjoy doing things in the outdoors, so hiking, backpacking, camping. And then I also really enjoy other creative pursuits such as art and music. I'm very lucky living close to New York City. There's a lot of opportunities to enjoy these things. I especially like to support emerging artists. So young artists and musicians, I like to go see their shows and encourage them, maybe sometimes collaborate with them. I think about creativity in art as being really quite similar to creativity in science. And I like to think about that and how these different fields run in parallel to reflect our society and new advances that we make.
Absolutely. And you mentioned collaborations. Are you an artist or musician yourself?
I'm not, but actually I'm collaborating with some bio artists. So recently I had several collaborations and I really like the way that artists think about science and also the way that they can make representations of science that make it much more accessible to the public. So I think that's really an interesting area that I want to explore more in the future.
Absolutely. I love these bridges between the worlds of art and science. And I think you're absolutely right. They can complement each other quite nicely. So it sounds like you've got a lot of different things going on outside the lab. Let's talk about your science next. So Dave, how do you describe what you do in the lab to someone who is outside of your field or perhaps outside of science altogether?
So usually I try and relate it to plants, obviously, because that's what I work on. And I tell people I just want to basically understand what makes plants grow and why different types of plants look very different from each other from a viewpoint of understanding how the genes work and how they control the shape of the plants. And if we get deeper into the conversation, then I talk about genetics and stem cells, which is something I'm very interested in. And most people can relate to that because of their importance in biomedical research. And then also about potential to improve agriculture using this very fundamental knowledge.
Fantastic. Well, I love how you describe your research and kind of bring it to where people are in terms of their interests and what their knowledge is about some of the different aspects of the work that you do. And we'll go into some more detail about some of your projects as we go through our conversation today. But I love talking about motivation in the beginning of these conversations because I think life as a scientist is hard, right?
Yeah.
So having motivational quotes and sayings and just these mantras in your mind can help you get through difficult times. So do you have a favorite quote or a saying, Dave, whether it's about science specifically or life in general?
I mean, one quote I like and often discuss with my colleagues or trainees is one which is fortune favors the brave. And I think it's very apt to science in that we do a lot of different types of projects as scientists, and some of them are sort of more routine, and some of them are, you might even call very risky or crazy, guaranteed to fail. But I think for people who are brave and do some of those very risky experiments, I think there's a certain potentially luck that comes with that we can make new discoveries and fundamentally change our understanding of science.
Absolutely. So is this something you try to incorporate into your lab every day, doing some of these brave, risky experiments?
Yeah, usually I try to encourage people to balance day-to-day experiments with taking risks and some of these way out ideas that we have.
Certainly. Well, it's wonderful to hear about this motivational quote, and I think you're absolutely correct. It has apt application to science. And I think beyond these motivational quotes, it can help to have people alongside you as you're working as a scientist to motivate and inspire you, I think, especially in the early stages of your career. So when you look back on your own career, Dave, are there particular scientists who served as mentors or role models or just these inspirational figures that you looked up to?
Yeah, I think I'd have to actually call out all of my mentors during my career. They were all quite different people, but all shared a passion for science that I tried to copy, and also teaching me how to keep a good work-life balance. As a first-generation student, I never really knew about that it was possible to work as an academic scientist and have a career in this field. And so having people who are doing this ahead of me and seeing their motivations and having them encourage me really helped me make this big step into something I'd never really dreamed of being able to do before. If I had to mention one specific person, it would probably be my postdoc mentor, Professor Sarah Hake from Berkeley. She was really very positive about everything. She could get excited about the smallest result in the lab. And that really taught me that science is not always about big discoveries, but it's the small steps, and we should enjoy those and celebrate them as much as the big discoveries that may only come, once every couple of years or something.
Absolutely. I love this emphasis of celebrating the small victories, because I think that is tremendously important and can buoy you when times are tough. And you mentioned work-life balance. I think this is such an important topic and important for scientists in the early stage of their career. When they're seeing all these people who are tremendously successful, you just sort of assume that they're working all the time. What really helped you establish work-life balance?
That's really hard to say, but I think I've always had a lot of interests, and I've always felt that working all the time is not productive. So I did something actually when I was a postdoc. I felt a lot of pressure to work a lot, and even during the weekends. And what I would do is I would actually set a reward for myself. So I would say, if I work for half a day on the Saturday, then I can go hiking in the afternoon or go to see some art show or something. And having that ahead of me then gave me the motivation to just go in and spend a few hours in the lab before I went off and did something else for the rest of the day.
Absolutely. Your own psychology experiments on yourself, right?
Yeah.
Well, I love that, Dave. And I'd love to hear a little bit more about your scientific journey and just how you got interested in science to begin with. I think everybody has their own individual story, and the paths are so different between people. So what first sparked your interest in science?
So I grew up in the north of England, which was the home of the Industrial Revolution. And when I was growing up, there were still a few factories left over there. And my father was an engineer who worked in those factories to fix the machines when they broke down. And he also had a workshop at home. So we would often tinker around at home, making things or breaking things. And I especially remember taking some of my older brother's toys and taking them to pieces because I just really wanted to know how they worked. And what usually happens is I would open up some musical teddy bear or something and end up with 100 pieces after I'd finished and had absolutely no chance at all about putting it back together again. So I often got into trouble for that. But really had this sense of wanting to know how do things work by taking them to pieces, which is sort of what we do as scientists. After that, this was around the time when biotechnology was really starting as an important field, and some of the first biotech companies like Genentech were forming. So my dream when I was thinking about going to college was to work in biotech in industry. And I applied to the University of Leeds because it was the only university in the UK at the time that was offering an undergraduate degree in biotechnology. But then in my third year, I got to join a lab and do an independent research project and just really loved the idea of being in an independent lab, having my own project and being able to guide the research I was doing. And so I switched then from the idea of going to work in industry to thinking more about academic research. So then I did my PhD, as you mentioned, at the John Innes Institute, which is a great institute working on the agricultural genetics. I then did a postdoc, and I was fortunate to meet my mentor, Sarah Hake. She came to a meeting in Norwich where I did my PhD, and I met her and really liked the science she was doing, and I liked her as a person, and she seemed like a really supportive mentor. So I made a big step of going from England to California to join her lab for a few years. always thinking that eventually I would move back to the UK and work in the UK. But then, of course, that never happened. And I think it didn't happen because I found this community of researchers working in maize genetics who were incredibly supportive, open about discussing their research, and really made a lot of lifelong friends and colleagues who work in maize genetics who'd really, I thought it would be hard to leave that behind.
Definitely. So what was it specifically about the research area of plant genetics that captivated you and turned you away from this planned biotech career and has really kept you in the field all these years?
Well, you mentioned field. We do a lot of field work, actually. And I think it's the realization that I can have a career where some of the time I'm in the lab, sometime in the office, but I also spend a lot of time out in the field with the plants. And that's really, as a law of nature, there's nothing better than spending a couple of weeks out in the field, not having to think about meetings or conference calls and things, but just being with the plants, trying to understand how they're growing and make genetic crosses. It's really a very diverse set of opportunities that I have to enjoy science in different ways.
Definitely. And you mentioned it was a big jump going from the UK to that postdoc in California. So what was that experience like for you? And perhaps what helped you get settled starting a research program in a new country?
The main thing is that because science is so international, even though I was moving continents, when I arrived in California, there were people from all around the world working in the lab and in the institute. So it didn't feel like I was in a different country because my experience in my PhD was also that I got to interact with people from around the world. And so it really was a different location, but still the sort of environment that I like, interacting with people from different countries and different cultures, and having that input, I think, in different ways of thinking about science is really stimulating.
Certainly. And I think as you're approaching the end of your postdoc, it's sort of an existential crisis for a lot of people trying to transition to a faculty position or a permanent position somewhere. So for you, what drew you to Cold Spring Harbor?
Well, I knew about it through the work of Barbara McClintock, who's a very famous maize geneticist who won a Nobel Prize for her work. So I knew of it as a very famous place, but I didn't really think about working there. But it just turned out, I was incredibly fortunate that around the time I was applying for jobs, a job came up there and I applied. I knew somebody already working there and they encouraged me to apply. So I'm kind of embarrassed to say nowadays because the job market can be incredibly competitive and very challenging for some people. But I kind of fell into that job without really applying for other jobs. So it was a great deal of luck, I would say, that I managed to end up there.
A great deal of luck and a fair share of talent, let's be honest here, right? And you've been tremendously successful over the years. Can you talk about maybe some of these key moments in your career there at Cold Spring Harbor Laboratory that really helped you become the scientist you are today?
Yeah, I think it's various things, but having great people working with me. So Cold Spring Harbor is a very prestigious institution, and I've had the luck of having really great colleagues who have encouraged me. We have several groups here working in plant genetics, and so we advise each other's research and help each other when we have problems with certain projects. But also I've been lucky to have really great trainees who have really done most of the work that we published, and they've been very creative in their research. And I think I've lived in sort of a golden age of genetics, I would say, in a time where we've had a lot of genetic variation in maize and a lot of what we call mutants, so classical genetic mutants that had very interesting effects on plant growth. But my career started in the age of genomics, where we can really now go into the DNA level and figure out what are the genes that are broken in these mutants and how we can use that knowledge to understand how the plants grow and develop.
Certainly. And I know you've been able to work on so many cool projects over the years, but can you give us a window into what's going on in your lab right now? Is there a project that you're working on at the moment, Dave, that you are just the most excited about and want to share with us?
I never like to say I have a favorite project because we have various diverse projects and they're all very close to my heart. Mm-hmm. But one I'm quite excited about is a gene in maize we started to work on a couple of years ago. And it has a rather strange name. It's called terminal ear. It's called that because it makes ears at the tip of the plant rather than on the side of the plant where the ears are normally made. But what's really interesting about that gene is that we found out that not only does it control the development of the plant, but it also controls how the plant responds to heat stress. So as you know, with climate change, that heat stress is a big problem for agriculture. And what we found is that this gene may help the plant balance its growth with its responses to stresses such as heat or drought. And that was really an accidental discovery we made through an experiment that gave a very unexpected result. And then we were able to follow up on that and show that indeed these plants are differing in the way that they respond to heat.
Very interesting. And it sounds like perhaps it's not desirable to have the corn ears forming at the terminal ends of the plant, but is there a way to kind of leverage the heat stress resistance or those characteristics without getting maybe the undesired positioning of the corneas?
One of the amazing things about science is that every few years there's a new technology that comes online that enables you to do things that you almost wouldn't dream about in the past. But there's this new technology called CRISPR, which is a way to edit the genome. And with that technology, we found that we can not only turn genes on and off, but also sort of like a volume control, turn them up or down to any certain degree that we want to. And so we can control one aspect of the phenotype of that gene, one aspect of its function without affecting the other one. So the idea is potentially we could make normal looking maize plants, which are better suited to respond to heat stress so that they produce better even under drought or heat wave conditions.
Well, I think that is tremendously exciting. And I appreciate you sharing some details about this project. And I think when you're working in science, like we alluded to previously, sometimes it's a lot of work and a lot of struggle to get to some of these exciting breakthroughs and successes. So we love talking about what happens behind the scenes, behind the successes that people often see. So, Dave, do you have a story of some challenge that you experienced in your career or a major failure that you had? And if so, could you talk a little bit more about how you got through that difficult time?
It's difficult. I mean, I think we have to try to remain positive even in the face of challenges, which happen not every day, but very frequently. For example, we submit research for publication to a journal. They often get rejected. Grants often get rejected. So really, you have to stay positive and try to work through that. Unfortunately, one of the most recent things that has been happening is that funding for science in the US, the current administration is proposing to cut funding for science. And that's really causing a lot of challenges and a lot of uncertainty, especially for junior scientists. So we really have to rethink about how we do science, how we fund it, and try to stay positive and driven by the important questions that we have rather than just thinking about these day-to-day failures.
Absolutely. I think it's important not to wallow in the failures and the struggles. You have to keep motivated, stay positive.
Yeah.
But I guess, do you have an example of maybe a specific paper or a grant that you were really excited about that didn't end up getting funded or that was rejected?
Actually, the story I told just earlier about this gene that we think controls the interaction between growth and stress responses, that project has been rejected 3 times for funding. It's not that the reviewers didn't like it. I mean, in fact, one of the times it scored outstanding, but unfortunately the funding level that year was not sufficiently high enough to fund it. So I just keep going. We keep trying to work on the project using resources from other funding that we have. And I'm sure that one day it will get funded.
Certainly. And I think getting over those failures can be difficult, particularly for early career scientists who may be listening. So for you, when you opened that email and you saw, wow, great score, but then later figured out great score was not enough, how did you respond or how did you kind of bounce back from that?
I usually try and take a bit of time off, like maybe go home and spend time with my family for half a day or something. Just try to not think about it too much. This is sort of funny, but one of the good things about rejections is they're usually quite slow. When we submit grants, it often takes 5 or 6 months to hear the outcome. So you kind of forget about all the hard work that went into it. So that's sort of one slightly better thing about it. But still, it is very disappointing. I think discussing with colleagues also, especially I like to talk with people outside of science who have the challenges, but maybe analogous challenges like working in business. Often funding priorities change. So it gives a broader perspective on how to deal with these things.
Absolutely. And I love the point that you made about time. I feel like time can heal a lot of stress and frustration and just disappointment, and having that built into the natural process of applying for funding and papers as well can be very helpful.
Yeah.
Well, I appreciate you talking about some of the challenges that you've encountered, but we love celebrating the successes in science as well. And you mentioned this really cool project that you're working on. I think this is definitely a success, whether it gets funded or not ultimately. But do you have another success story you'd like to share with us, whether it was one of these big victories or even just a small but particularly meaningful success?
A couple of things recently. I mean, one is that as a group leader, I don't really do much science myself anymore. I'm more in a supervisory role, but I'm always really happy when my trainees have successes. So my graduate student, Matthias, went to a conference in the summer and he ended up getting an award for the best poster at the conference, which I think is a great testament for the science he's doing. And also just seeing him interact with other scientists at the meeting and being very interactive and asking questions was really inspiring to me that he was a good role model for young scientists. So that was really great that he got that recognition. And then also a small experiment recently. We're working on another project about how genes interact with each other in plants. So Normally you expect if you knock out a gene or if you disrupt a gene, you will see some kind of effect in the growth of the plant. But often that doesn't happen because genes often have backup copies. We call them paralogs that can step in when the first gene is knocked out. And these genes are on different chromosomes, but they're talking to each other and compensating for each other's activity. And we're trying to understand how that works. And there's already quite a bit of work in this area in the animal field where they think that there's communication between genes at the RNA level. And looking at different mutations, they figured out what kind of RNA may be communicating. But we've recently, using CRISPR technology, we managed to make a complete deletion of one of the genes. So the gene is completely missing now from the genome. So it's not going to be making RNA. So we're very excited to see what effect that's going to have on its sister gene, which normally would be compensating. So yeah, that's something that, again, 5 years ago, we wouldn't have thought of being able to do that. But with CRISPR technology, now we can go into the genome and make these targeted tweaks that can really be very informative.
Very cool. Well, Dave, do you have hypotheses of what you think might happen? Do you think it will mirror pretty similarly what happens in the animal research?
I don't like to predict these things, but my hunch is that it may be different. Some preliminary data we have suggests that there will still be compensation. In other words, that probably the RNA is not involved. So yeah, I think that's really interesting. And people don't often think about doing fundamental research in plants, but we should remember that a lot of important discoveries in genetics, including the discovery of genetics itself, was made in plants, of course, by Gregor Mendel. And so plants are a really great system for answering these fundamental questions in biology and gene regulation.
Certainly. Well, thank you for talking about some of these successes coming out of your lab. I think absolutely the accomplishments of your students can be tremendously rewarding. And also, this cool project sounds like it's really taking shape, and potentially listeners will have to keep an eye out for the exciting findings coming out soon.
Yeah, I hope so.
Certainly. And I think as a scientist, having these exciting findings can motivate you to get into the lab and work harder and harder and focus more on the science. But we try to encourage our listeners to take a break and to perhaps pick up a good book to give their mind a break and think about something different. So we love getting book recommendations from everybody on the show. So Dave, do you have a favorite book or one that you've enjoyed recently that you'd like to recommend, whether it's related to science or not?
To be honest, I don't read a lot of books because I spend a lot of my time in the office reading papers and grant proposals and things. So when I'm out of the lab, I prefer to have other outlets like music. But one book I think any young person interested in science should read is the biography of Barbara McClintock by Evelyn Fox Keller. It's called A Feeling for the Organism. And I think it's a really great account of how a scientist can be passionate about their work and through very careful observation can make really fundamental discoveries, not only about plants, but also about all living organisms. And I think especially being about a woman in science working in the 1940s and 1950s when that was not so common, I think it's very inspiring to hear the stories of how she was able to make such fundamental discoveries about science.
Certainly. Well, I love this recommendation. I will add it to our website for our listeners to find there. And you mentioned that you also have these interests in art and music, and perhaps that's your preferred outlet when you're escaping from science. Can you tell us a little bit more about what kind of music you like or what kind of art you're into these days?
Musically, I really like jazz. I like music that's quite unstructured. So I've been listening to a lot of experimental jazz. For some people, it might just sound like noise, but to me, it just resonates somehow. Especially, I am fortunate to know a few musicians, and recently we had a jazz concert at our house, and I was able to invite my lab members to that, and I think they enjoyed it.
Oh, cool.
Yeah, it was really great to support these young musicians in their exploration of music. So that was really fun. And on the art side, as I mentioned, I've been exploring bioart. I'm a member of a citizen science group in Brooklyn called GenSpace that brings together a lot of people from outside of science who are interested in researching biology. And so I've had the opportunity of interacting with some bioartists and have collaborations. Actually, in corn, I'm collaborating with one bioartist who's interested in the origins of corn. He's from Mexico and very interested in the cultural implications of growing this crop from Mexico that has a lot of spiritual meaning to the Mexican people, and various other collaborations that are kind of fun to think about science in a different way.
Well, it's wonderful to hear about these diverse interests that you have outside of your science. And I think that goes contrary to some of the stereotypes that people may have about scientists. I think there's this picture in people's mind in the general public, or often in the media, how we're portrayed, just of what scientists are like, wearing their lab coat, being very serious, only thinking about and talking about science. And I think that's just not the case for so many scientists that I've met. So we love trying to break these stereotypes that people may still have by talking about the different personalities that you see in science, the quirks, the fun traditions, and just the funny memories that you share with colleagues from your lab. So do you have examples, Dave, of whether it's quirky traditions or just fun or funny things that you've done together over the years with your fellow scientists that go against some of the stereotypes people may have?
Sure. Yeah. Maybe not quirky, but I have a tradition of bringing my lab to New York City around the holiday times to go ice skating. And I think it's really fun to interact with people. We're used to discussing with our colleagues and being very serious talking science, but just to interact with people when they're ice skating, many of them have never been on ice skates before. And it's really funny to see their reaction, and many people fall down and we really have a good time. I remember I had a postdoc researcher, Jocelyn Benitez from Cuba, and naturally she's a really good dancer. And whenever we went to meetings, if there was a party, she would embarrass me by dragging me onto the dance floor because I'm a pretty terrible dancer. But then I enjoyed bringing her to ice skating and figured out she couldn't ice skate and fell down a lot. So we often joke about that even now.
Oh, I love that. Are you an accomplished skater?
Not so good, but over the years, I can at least stand up for most of the evening. Yeah.
Good. And if you fall, it's just to show sympathy for your fellow skaters, right?
That's right. Yeah. Yeah.
Well, I love that. And I think this is a wonderful example of just getting people outside of the lab environment to get to know them better, to build that camaraderie. And I think in addition to that, when we were talking about just your experiences and your academic training, we highlighted some of the opportunities that you've had to travel, to go to different places for your work. And I think this is something that expands even further once you're in a faculty role, going to conferences, working with colleagues and collaborators around the world. So let's talk a moment about scientific travel. I think this is a perk of the job, Dave. So do you have a favorite place that your science has taken you, or perhaps a most memorable travel experience you can share with us?
As you say, it's a great opportunity as a scientist to travel a lot to conferences. Perhaps my favorite place I traveled to is Japan. I first traveled there as a postdoc. My advisor was invited to a conference and she couldn't go. And so she asked me if I would stand in for her. And I was very happy to do so. And I just really love Japan. Not only as scientists are extremely creative, but also really interesting culture there and countryside. So I'd spent 2 weeks traveling around Japan, exploring the culture, but also visiting different labs, and made friends and colleagues who've become lifelong friends. So it's been very motivational to me to have that connection with different cultures and scientists. One thing that I think people don't understand is that in science, we become extremely focused on specific topics sometimes. The project I described earlier on this gene that interacts between growth and heat stress, the only other lab in the world working on that gene happens to be in Japan. And so I have a really good opportunity to visit them recently, and we can discuss our progress and help each other out in making progress. We work on the same gene in different plants, but really the basic principle is the same. So yeah, that's really motivational to have colleagues around the world who are really interested in what we're doing. And obviously I'm very interested in what they're doing also.
Certainly. Well, this sounds like a fantastic collaboration. And it sounds like you had a wonderful experience being able to go to Japan for the first time. Was it intimidating having to step in for your advisor for this trip?
It was quite intimidating because it was my first international conference. Mm-hmm. And also I didn't realize before I went there, but all the talks, apart from my talk and one other talk from an American scientist who was there, all the talks were in Japanese. So that was also quite challenging. But actually, I think I gained some kind of amusement among the attendees, because even though the talks were in Japanese, I was still able to ask questions because, of course, the pictures I could understand. And so they thought it was funny at first that I was asking questions, even though I spoke no Japanese. But in the end, it made sense.
Well, I love that. And you mentioned you were able to travel around Japan for 2 weeks. What were some of the places you went to?
So I started in Tokyo, and then I went to Kyoto and Nara and Nagoya. So mostly in the central island, Japan. But since then, I've been back many times and explored many different areas, parts of Japan. So yeah, I've seen quite a bit.
Very cool. So if listeners are able to go to Japan sometime soon, what is a must-see thing they should put on their itinerary?
So many things. One thing to know is if you go in the summer, it's extremely hot and humid, especially in the cities, can be quite oppressive. But this summer I was there for a conference, and afterwards with some friends, we went to the Northern Alps, and that was really beautiful. I spent time in the European Alps, but it's really spectacular mountains, very easy to get around because the public transportation there is so good, and great opportunities for hiking and enjoying the outdoors. I would really encourage people to explore the countryside as well as going to the obvious big cities like Kyoto and Tokyo, which are also very impressive and stimulating in different ways.
Definitely get out, see some of the natural beauty while you're there. I love it.
Yeah.
Well, Dave, thank you for sharing some of these fantastic travel opportunities that you've had as a scientist. And I think the work that you are doing is tremendously important. You're answering some fascinating questions with your research, but oftentimes, as we alluded to previously, things like funding can be a limiting factor. Also, just technology, staff, feasibility, even time. So if we took away the things that typically hold you back in the lab and we gave you everything you could dream up, what is the one research question, Dave, that you would most want to answer?
There's a very basic question in developmental biology that people often state as the motivation, which is, could we take a single cell and convert that into a whole organism, like regenerate a whole plant from one cell? And we've learned so much over the past couple of decades about genes that control plant growth, but I think we're still a long way away from addressing that one question. Can we take a cell, turn that into an embryo and turn that into a plant? And I think that plants do this every day. This is happening billions of times every day inside seeds, but it's still something that we're not able to work on to program in a lab. So yeah, I think with unlimited resources and some ideas and good colleagues, I think it would be a really important problem to tackle.
Certainly. So what do you see as, I guess, the biggest barrier? Is it just sort of a bandwidth issue? Is it you don't necessarily have the technology to do the things you need to do?
I think it probably needs a combination of a lot of different types of technology. So not only the genetic knowledge, but also knowledge about the environment in which the cells grow. So the biophysics of that and the culture conditions. So yeah, really a lot of different things. And the insights will come from genomics, I think, but also being able to understand genomic data. One of the problems these days is we have so much data, but understanding it is probably something that will require artificial intelligence, which some groups are working on that in biology, but I think it's an area that still needs a lot of refinement and improvement to really gain deep insights into how cells are formed and how they are programmed.
Well, Dave, this is a fascinating research project for me and our listeners to ponder today. We appreciate you sharing it with us. And you've shared a lot of different things about your career and your work with us today, but I'd love to end by talking about advice. I think for our listeners out there who might be at the crossroads in their career or just in a difficult moment, advice can be tremendously helpful. So do you have one piece of advice that somebody gave you that really helped you that you can share with our listeners today?
Just not being afraid to try things. As I mentioned earlier, when I was an undergraduate and thought I knew what I wanted to do, and my advisor said, just try working in a lab for a year and see if you like it. I think sometimes we're sort of so concerned about progressing in our careers and think there's a timeline we need to fit to, but there's nothing wrong with taking a year out and trying something different or trying something that you're not sure if you will enjoy it. But for me, spending a year just in a lab without any deadlines or degree I was applying for, I was just researching in a lab and then really figured out that I really enjoyed it. Science is an amazing career and there are many different ways to enjoy it, but taking some time to explore different areas can be very useful.
Absolutely. I think that is fantastic advice. And I think particularly today, there does seem to be just that pressure to sort of produce quickly, to always be moving along the pipeline, making these papers happen and just moving right along. But taking a break can really help.
Right.
Well, fantastic advice for our listeners. Is there any other last note of inspiration or motivation that you would like to leave them with today?
I think we're at a real inflection point in biology. I think there's some incredible new technologies emerging, like CRISPR, genome engineering. And also, I think we have some really important challenges, including climate change and in biomedicine, for example, some diseases that with these technologies, there's a great opportunity to make really big discoveries and really improve people's lives. So I think it's a really great time to enter biology. And for anyone who's interested, I encourage you to reach out to colleagues, reach out to professors nearby, and try to join programs where you can learn more about these new technologies.
Absolutely. I think it's a mix of a lot of exciting technologies and a lot of possibilities, but also a lot of big problems, unanswered questions, and tremendous challenges at the moment. So
that's right. Yeah.
The perfect stew. Well, Dave, a wonderful note to end on. If our listeners want to learn more about you and the work that you're doing, what is the best way for them to do so?
I have a website. So if you search for Jackson Lab at Cold Spring Harbor, then you'll find my website. It has links to the different projects that we're working on, also links to some of my bioart collaborations. And so, yeah, I encourage people to take a look at that and also contact me. I'm always happy to discuss with people about our research and learn more about other people's research too.
Perfect. Well, listeners, definitely take some time to peruse the lab website and take some time to reach out if you have specific questions for Dave. And Dave, it's been such a pleasure to chat with you today and to learn more about you and your career.
Thanks so much. Yeah, I really appreciate the opportunity to spread some of the word about our science, and hopefully will inspire young people to follow this career also.
Absolutely. Well, we appreciate your time today, listeners. We appreciate you joining us as well. And we hope you'll join us again for our next episode of People Behind the Science.